So today's going to be another pretty casual stream. Cell biology is something that I hold near and dear to my heart. I've lost track about how many times I've had to go over the different parts of cell, whether it was in school or like in post-secondary with my courses. That said, this is a very, very deep rabbit hole. There are parts to parts to parts, going down to interactions at the atomic level to doing things at the biochemical level, where you're specifically looking at how like proteins might interact with like other proteins or attract molecules and stuff like that. So cell biology is fascinating. It's also something everyone should look into, especially based off of the last couple of years. When you learn about cell biology, you basically learn about how these really tiny interactions can affect like the whole organism. Like it only takes the binding of a certain chemical to a receptor to cause a whole bunch of downstream effects, especially if what's binding to that receptor can affect multiple cells over its lifetime. An example of that would be something like, what was it? Botulinum toxin? Yeah. That one, but it has a really big potency, not just due to its downstream effects, which causes like paralysis or even death, but the fact that it takes a while for it to be degraded, hence why it's regulated so strictly. Let's see. Anyways, so this stream is going over the anatomy of the cell. I'm only going to go through a few organelles and maybe make some annotations here and there. I'm also going to see how far I can get from memory alone. If I'm unsure about something, I'll probably just not talk about it, just to keep it more clean, I guess. Anyways, without further ado, let's get drawing. So over here I have a few organelles. I'll start with the animal cell first and then go to the plant cell, because there are a few differences between them. I won't be doing fungi or prokaryotes, because those are different. I also won't be doing archaea, because that's also different. That's like whole different... Is there a domain of life? Yeah. Yeah. Microbiology is so cool, but also it's such a vast field. Like, there are innumerable amounts of bacteria being discovered every single day, and it's just gonna keep going due to like how they develop evolutionarily wise. It's a miracle we can track anything at all at this point. But anyways, let's get started. So, an animal cell, overall, kind of looks like a blob. Under microscope, it's typically transparent too, but you can use different dyes in order to, how do you put it? In order to increase the contrast between the background and the foreground. Typically, when you look at it under a microscope, say it like, I know, 10 times, you're going to be able to see the outline as well as the nucleus. After that, you're going to need to use dyes in order to make other things more apparent. Either that or use a stronger microscope, like oil-based with 1000x, or maybe even like a transmission electron microscope or a scanning electron microscope. But those usually aren't available until the university level. Although they are really cool, especially scanning electrons. Oh my gosh. Like, the quality of the images and like the textures from it is incredible. Anyways, so from here, you can sort of see the cell membrane. and then also the nucleus. So the cell membrane is this, how do you put it? It's this barrier that controls what goes into and out of the cell. It keeps the cell's contents inside and separates it from the outside environment. Without the cell membrane, you'd basically have no cell because the insides would leak all over the place and the cell would pretty much be dead. So there's certain ways that molecules can pass through the cell membrane. Usually there's like 3 different types. Either it like diffuses over, so let's say we have like a bunch of dots over here, and a few of these dots over here. If they're small enough and if they are charged, like, I know, water for instance, or oxygen, it'll go from the higher concentration over here to the low concentration inside. But again, only if it's like, how do I put it? That's only if it's like small enough and somewhat charged. Meanwhile, the other 2 types are like active transport and facilitated diffusion. So let's say over here, right? If we were to zoom in a little bit, the cell wall, or the cell membrane, has essentially 2 layers to it. A phospholipid bilayer, which kind of looks like this. And then embedded within, you'll have these carrier proteins, as well as other proteins used for active transport. So color it sort of like that, and that can be like that. So with I put it, with this sort of bilayer over here, you'll have a charge outside, and then with the phospholipid tails, these create a nonpolar inside. But I guess I'll kind of go for that later. With facilitated diffusion, though, it kind of follows the whole thing where you go from high to low, only this time, if you have a big particle like glucose, the glucose can pass through here, and then the transport protein will change shape, allowing it to go from high concentration on the outside to low concentration on the inside. Active transport is sort of similar, only it needs energy. It's like ATP in order to power this whole system. Without ATP, you don't get that transporter working. There's also something called protein channels, and those are used to transport certain ions from high concentration to low concentration. With channels, they don't have the ability to be turned off, typically. Or no, wait. Sorry, there are channels that can open, and then there's other channels which are always open. I'm trying to remember. I believe a few of those channels are used in nerve, or like during an action potential in nerve cells, but I'm not really going over that today. Anyways. Let me just get rid of it. Actually, no, that can stay. Let me add another layer. Put that away. Okay. So, let's go over a few more of the organelles. So, oops. Within your cell, you're going to have this jelly-like substance called the cytoplasm. Yeah, that's fine. So the cytoplasm is typically transparent, mostly because it contains a lot of water and nutrients. And this is important because it'll suspend all of your organelles inside, so they'll be like floating. Additionally, because it's nutrient rich, it allows different organelles to use those nutrients in order to power the metabolism and like undergo different biochemical reactions. This is pretty nice. Okay, after the cytoplasm, there is the mitochondria. So these kind of look like jelly beans within the cell, like that. And a cell can have multiple mitochondria, it's not just one. You'll find the most amount of mitochondria in a cell in something like the muscle cells, because the mitochondria is basically the powerhouse of your cell. It'll convert glucose, or rather, no, it's pyruvate actually, to be more specific. It'll convert pyruvate into basically a lot of ATP and other high energy intermediaries, and you use that ATP to, help out with transport around the cell, to help out with other forms of metabolism. It's just really, really useful. Anyways, so the mitochondria is also rather unique because you inherit these from your mom. You don't get any of them from your dad. What else is there? I believe they have their own nucleus as well, but sometimes it can be kind of hard to see. Yeah. So the fact that it has its own nucleus, the theory behind it is that the mitochondria was once its own original cell, and that it got swallowed up by the precursor cell to, plant and animal cells, but the mitochondria didn't die. Instead, it was like, hey, it's kind of cozy in here. Let me chill out for a little bit. And, because you're being so courteous to me here, let me pay up my rent in ATP. I'm really good at producing that. So they went in, or the mitochondria went in, and basically it's been with us ever since, just producing a lot of ATP for us. Kind of nice, actually. Alright. Let me just move a few things around. Oh. Yeah, that's good enough. Okay. Next up, lysosome. Actually, I might as well do lysosome and vacuoles and vesicles all at once. So the general structure of a lysosome kind of looks like that, like a blob. But within the cell, lysosomes are very hard to see. They basically look like dots. And you definitely need to stain the cell in order to see them. Otherwise, they're really, really small. Same thing goes for vacuoles. It's different for plant cells, though, with how their vacuoles are structured. But the reason why I kind of want to group these two together is that they're both pretty close in terms of structure. What varies, though, is what they carry around. So they typically look like this. Actually, hold on. Yeah, there we go. Need to decrease my pen size. There we go. So you might have seen the structure before. And that's because vacuoles and vesicles and lysosomes are essentially pieces of the phospholipid bilayer that have formed a sphere and that contain stuff on the inside. So they'll have proteins that act as like cell receptors on the outside, and then a gap in the middle where they store stuff. So they'll have like nutrients. But in the case of the lysosome, the inside over here will contain a bunch of catalytic enzymes. And these literally break down anything inside. So for instance, if a bacteria gets caught in there, the catalytic enzymes will attack it and cause that bacteria to die off normally. There are a few examples of bacteria being able to get out and evade that, though. I'll have to double check my notes and maybe prepare that for another stream. Yeah. But these lysosomes over here, they don't just attack bacteria, they're also used to take out old and dying organelles as well. So think of it like the garbage man of the cell. Without these, you'd have an accumulation of old, decayed structures that would take up way too much room inside the cell and couldn't be used for anything. Alright, next up. I should probably discuss vacuole as well. Hold on. Vacuoles are also containers. They have a similar structure to lysosomes, but they don't just carry catalytic enzymes. They're normally used to move things around, such as they're normally used to store things like water and other nutrients. If a vacuole is in motion, it basically turns into a vesicle, and vesicles move things from one site to another. I didn't put it on the list, but it's not too big of an issue. Alright, next up, rough endoplasmic reticulum, smooth endoplasmic reticulum, and Golgi apparatus. So these kind of all go together. So here is our animal cell. Here is our nucleus over here. And then branching off from that, you'll see something like this. Kind of looks like a maze in some aspects. And then some parts of it will be smooth, but the other parts will have these bumps on it. And then just off to that, you'll see something that looks like pancakes or flat discs, like that. Okay, so I'll explain this part first. This over here is your endoplasmic reticulum. I'm not going to write it out, I'm just going to shorthand it. And it has two components. If it's smooth, like what you see over here, this part is called the smooth endoplasmic reticulum. It's used mostly for like synthesizing fats. Meanwhile, if you have something over here with the bumps, those bumps are your ribosomes. And these are responsible for producing proteins. So proteins are a very broad category of macromolecules. They can be used for anything from like binding to cells and doing cell signaling to acting as like structures for stuff like keratin, which makes up your hair and your nails. Just a ton of different ways proteins can be used. Anyways, so it's pretty easy to spot the endoplasmic reticulum because it's very close to the nucleus. And how do you put it? And that's pretty important because like the reason why these are so close to each other is that it makes making proteins really efficient when you do transcription inside the nucleus, translating that DNA into mRNA, and then translating that mRNA instructions into proteins once you hit the rough endoplasmic reticulum. Then those proteins get transported by vesicles from the rough endoplasmic reticulum over here to the Golgi. Golgi? Golgi. I can never get it right. So there's different parts to the Golgi. I believe there's 3 different parts, but I need to double check. The parts are basically based upon where the Golgi apparatus is facing. So they'll enter through one side, get packaged, and released to the other side. So vesicles come in, the proteins get packaged and folded, and then exit and get sent along certain actin. Is that actin? Yeah, along certain highways along the cell. They don't just get sent off randomly, they have destinations where they go depending on where or what contents they have. Typically, they usually get trafficked to the cell membrane, though, so that they can get exported or become part of the cell membrane. Yeah. So think of the rough ER as like a manufacturing plant where they take the blueprint, that mRNA, and turn it into actual products, your proteins. And then think of the Golgi as your packaging plant. All right. Next up, we have the nucleus. So the nucleus is basically kind of like this. Yeah, it's basically a really dense circle that contains your DNA. Now your DNA is typically supercoiled, so it's all like squiggly and stuff like that. And it's contained within a nuclear envelope or a nuclear membrane. And that's very important because the nuclear membrane controls what enters and leaves the nucleus. Let me just do it like this. Yeah. Without that membrane, it would be very easy for viruses to come in and hijack like the mechanisms for DNA replication as well as as well as DNA repair. It doesn't block everything, but it blocks a lot, which is good. Yeah. I'd totally love to talk about viruses in a stream, though. I just found it pretty fascinating. It's been interesting to see their portrayal in stuff like science fiction, whether it be like in a zombie apocalypse with like T virus, B virus, that's everything in Resident Evil, as well as how it's portrayed in like science fiction. Oh well. So the nucleus acts as like the brain of your cell. It stores your DNA, which acts as like the blueprints for how to create like an organism. So it's important for like, how do I put it? Typically this DNA unwinds. It goes from this jumbled mess to like chromosomes later on, like that and like that. But that's typically during DNA replication. What else is there? Yeah, And these are also, the DNA is used in something called transcription. It's a step where an RNA polymerase, like a protein, attaches to the DNA. So your DNA kind of looks like this. Yeah, there you go. The DNA polymerase will come in like this, attach to it, and basically start reading and create this mRNA script. So this mRNA script or blueprint exits the nucleus, gets modified, and then gets used as a blueprint to create proteins with the ribosomes on the rough endoplasmic reticulum. Fun stuff. Alright, I think that's about it for the animal cell then. Overall, it's not too bad. You just have to remember what each of the different functions are. Usually people kind of mess up with the smooth and rough ER. Let me see if we can find it. Nope. Yeah, there we go. Just remember the smooth ER doesn't have the bumps. Instead it's used, because it doesn't have those bumps, it's kind of slick like fats. Then the rough endoplasmic reticulum is all bumpy due to those ribosomes, and it's used for proteins, like protein synthesis. Alright, so now we can move on to plant cells. So the first thing that you'll notice with plant cells is that they're not blobs. They're typically more rectangular or squarish, and that's because if you look over here, they have something called a cell wall. Let me put up my other list too. Yeah, there we go. So, like animal cells, they have their cell membrane. But above that cell membrane is your cell wall. So the cell wall isn't unique to plants, it's also found in bacteria as well. But bacterial cell walls have a different structure than plants. They both, how do I put it, the cell membrane and cell wall both have the purpose of providing structure to the cell. But the cell wall in plants greatly improves the plant's ability to be like rigid and straight up. Without that cell wall, plants would not be able to grow up to like the major sizes that you see down in with like the California redwoods or stuff like that. So this rigid strength over here allows the plant to build up to much, much bigger things out there in the real world. For animal cells, in order to reach that sort of size, you need to be like suspended in water, because otherwise your body would literally collapse in on itself, which is not a good thing. Alright. So plant cells have pretty much all the same organelles as animal cells. They still have cytoplasm, mitochondria, lysosomes, rough and smooth ERs, Golgi's, nucleus, which you can typically see. But they do have a few unique features to them. So there's the cell wall. They also have a large central vacuole, which I'll represent in blue, like so. This is important because this mainly stores water. And it doesn't necessarily appear blue, it's more like transparent. But basically, as the plant cell gets more and more water, the cell will expand and the cell membrane will push up against the cell wall, making it turgid. And that's a good thing for a plant. It wants to be turgid so that its leaves won't droop and it can maintain its structure. What else is there? The other important part for a plant is the fact that it has chloroplasts. You can probably tell from the name what the purpose of those chloroplasts are. So they're green, like actually green, and they're used for photosynthesis. So, central vacuole. Chloroplast. So the chloroplast is used for photosynthesis. This is how plants make their own, or use the energy from the sun in order to make their own food, glucose. Without these chloroplasts, they'd be really similar to animal cells. Additionally, without plants, there probably wouldn't be life as we know it on Earth. We'd probably still be in like very, very basic forms, or like huddled around like a like a heat vent underneath the ocean in order to get our food. So with chloroplasts, they convert CO2 plus water into glucose, C6H12O6, as well as oxygen. Get a piece of that oxygen, that layer. How's that speech go? Be a high-tier human. Yeah. Yeah, and that's basically all the differences that you generally see. So again, they're less blobby and more shapely, like rectangular-like, and that's because of their cell wall. They have a large central vacuole, which mostly stores water, and getting more water makes that cell turgid, which is good for the plant. And they also have chloroplasts in order to photosynthesis. Oh, you can't see that from there. So chloroplasts equals photosynthesis. And a plant cell can have more than one chloroplast. They're actually able to move their chloroplasts between different plant cells, which is really cool, actually. We call that cytoplasmic streaming. Anyways, what else is there? Yeah, I think that's about it for the differences between those two. Yeah. I guess I can go a bit into bacteria since I have a bit more time than I thought. This is just extra notes, like university level, because I don't believe they teach this stuff in high school, which I guess kind of makes sense since it's mostly based on like human body systems, but it's also important to learn how bacteria work because, how do I put it? I mean, not to trigger any germophobia or something like that, but your body is covered in bacteria. I mean, not just covered, but they inhabit a whole bunch of different regions of your body. Like, literally trillions. They inhabit stuff like your skin, of course, inside your mouth, which, if you don't keep under control, can lead to bad breath. In your gut, where they typically provide, and like, they digest certain types of food from your body and provide back nutrients. Other times they can act as a barrier and crowd out bad bacteria, but other times if they end up in the wrong place, they can cause certain health issues. What else is there? You have parts of bacteria which can end up in your lymph, which can be used to train your immune system in order to recognize what's supposed to be there versus what shouldn't. I'm pretty sure you can have... No, never mind about that part. Yeah. Typically, bacteria can be organized into two separate categories, where you have gram-positive and gram-negative. And we call it that because there is a technique called a gram stain. So the bacteria will turn a certain color if they're gram-positive, and if they don't have, or if they're not turned, they're called gram-negative. And that's because this, due to the differences in structure of their cell wall. So with gram-positive, they have a really thick layer. Meanwhile, for gram-negative, they have really thin layers, like that. So in a gram stain, the dye will, I guess the dye kind of looks like, the dye particles are able to attach onto that thick layer that the gram-positives have, but they're not able to really attach onto the gram-negatives because it's too thin over here. There are many different tests which you can use to distinguish between bacteria. Typically, you grow them on different types of petri dishes, sort of like, so you'll have your petri dish like this, and it'll contain different nutrients on it, like blood. Oh yeah, that was a fun one. Blood agar. You have like a regular petri dish. Let me just draw this out. Yeah, so wait. Yeah, your normal petri dish would look kind of pale like this. Pale yellow. And then you might have blood agar dishes. What else is there? There are a few other types, but this is typically the simplest way of doing so. Just like have different types of food in the dish. If the bacteria grows on that, it means it's able to metabolize those ingredients and live. If it doesn't have those processes, it'll die, like straight up, and starve. And then typically what happens is if you want to see what bacteria is on like a sample, like a contaminated surface or something like that, you would get a swab, you would put that swab into like a test tube, kind of like that. Yeah. You'd swish it around. And then you would apply that onto your dish. And then you would streak it in a certain way. So you'd go, is it 3 different areas? Yeah, kind of like that. So you'd go 1 layer over here, make a bunch of lines, then take that's okay. Then take another layer over here, go down, do a bunch of lines, and then you go from here all the way down, do a bunch of lines. Something like that. The idea being that your diluting your colonies until eventually you can pick out one colony at the very end. So eventually it should look, how do I put it? So when you streak it out, you'll end up with a lawn on the first part, where it's really hard to pick out individual colonies. But then as you go further, the colonies will start to spread out a little bit, until eventually you can pick out single colonies, meaning that they come from the same bacterial cell instead of multiple ones. And you don't need like 5 or 6 to be separated out. Sometimes you just need one colony to survive and be isolated. Yeah. But that's about the end for this stream. Hope that helped your appreciation for biology. Yeah, biology is something that takes a lot of practice getting used to. It's not like physics or chemistry where the concepts are straightforward and you can use like formulas in order to work your way around things. It's more about like seeing how different things are related to one another, as well as creating experiments where you can control like confounding variables and finding like good results. And there's fields within biology. It's not just one big monolith. There are many different layers, whether you want to go down to the microscopic level and work with cell receptors. You could also work with the cells themselves, or move up to the entire organism, or even beyond that, and work with the entire ecosystem itself. So seeing how different animals interact with the environment, and how changes in the environment can affect them. But I think that's a story for another time. Well, I think that's about it. Hope you guys have a good night.